Hello, this is Dr. Moy. This represents the second educational video designed for the layperson to better understand cancer research and the challenges that underscore the difficulties in conquering cancer. In this video, I will discuss the cellular constituents of cancer, which is necessary to understand why it is so difficult to eradicate cancer with drug therapy. From session 1, we discuss there are significant limits in chemotherapy in curing cancer, and such therapy represents significant healthcare costs. Cancer drug development has the highest drug failure rate. The consequence of years of research has led to fundamental knowledge gaps in cancer biology that are preventing successful drug development. The historical research approach in drug development is to conduct basic research in cancer, identify lead drug candidates, test safety in animals, followed by final formal testing in clinical trials. Now, if all of these steps prove successful, then the drug is finally available to the general public. As we have previously shown in the prior session, the cost of drug development is expensive and prolonged, and the failure rate is extremely high. In effect, cancer research is skewed to trying to find treatments for the patient that will be diagnosed 15 years from now with little probability of success, while current cancer patients are typically neglected from the benefits of medical research expenditures. This raises the question whether are we missing an important opportunity in not integrating current cancer patients into medical research? More specifically, could society improve drug development if far more current cancer patients are participating in medical research? The notion that drug therapy has historically failed to cure cancer is best illustrated by the historical trends in cancer mortality. In 1986, Dr. John Baylor III published a critical report in the New England Journal of Medicine that examined cancer mortality between 1950 through 1982. To the disappointment of the cancer community, he demonstrated that cancer mortality didn't change between those years, concluding that the war on cancer was a complete failure and society should instead focus more on prevention. A follow-up report by Dr. Baylor in 1993 showed that cancer mortality was actually increasing, again reinforcing the need to shift the focus toward prevention and early detection. The National Cancer Institute created the Surveillance, Epidemiology, and End Result Program, or SEER, which accumulate ongoing cancer statistics. In the adjacent figure, cancer incidence and mortality is depicted for men and women broken down for African Americans and Caucasians. As shown in the figure, cancer mortality does not start declining until after 1995 in both sexes and in both races, not so much from medical therapy, but largely attributed to decreased smoking and greater focus on prevention and early detection. Also, the incidence of cancer is declining in men, but the incidence of cancer is increasing in women. If medical therapy has not historically cured cancer, then we need to review the history of medical therapy and figure out what we have learned. Cancer treatments have seen a variety of cancer drugs as a result of various scientists with different expertise migrate into the field. In each case, there has been initial excitement and hype about each class of drug introduced only to result in subsequent disappointment. After the disappointment settles in, the cancer community rarely spends much energy on why cancer drugs fail, abandoning the current research only to move on to the next big thing. The following scientists have introduced different classes of drugs, and this include chemists who have introduced cytotoxic small molecules. This remains the mainstay of chemotherapy for decades. Vascular biologists introduced vascular inhibitors with the rationale that by choking off the vascular supply of cancer, the tumor would regress. However, this has not resulted in universal success. Virologists introduced antiviral vaccines. However, this has only proven successful for viral-mediated liver cancer and cervical cancer. Protein biologists introduced antibodies like Herceptin that could inhibit breast cancer growth. However, many subsequent antibodies have failed to achieve the same level of success as Herceptin. Geneticists focused on targeting cellular pathways and cancer gene targets with inhibitors like Gleevec, which has been a major success for treating chronic myelocytic leukemia. Yet targeting approaches has not been universally successful for many other cancers. Immunologists have introduced vaccines and cellular therapies. While it has been beneficial for melanoma and renal cancer, such therapies have yet to be universally transferred to other cancer. It is safe to say that the jury is still out on this class of drugs. Nevertheless, the cost has been extremely expensive. Few new cancer drugs get approved, which illustrates the complexity of cancer. For the majority of patients, we are still using the same cytotoxic chemotherapy introduced decades ago. Over the past decade, there has been strong interest in genomics and developing drugs based on targeting the genetic aberrations and metabolic pathways of cancer. However, the effort has proven very challenging. Further, new drug targets that are approved have been extraordinarily expensive compared to traditional cytotoxic chemotherapy, often resulting in an improvement in survival of only a few months. A common problem in cancer therapy is the phenomena that cancer exhibit significant drug resistance. Every drug class has hit the same wall and has not moved the needle in curing cancer because of this drug resistance and a lack of drug efficacy. Thus, there is a fundamental knowledge gap in understanding drug resistance and predicting drug efficacy. To begin to understand the issue of drug resistance and efficacy, one first needs to understand how drugs are identified in the first place prior to being introduced into a clinical trial. This process is depicted in the slide. First, an artificial cancer model is required, which is representative of a patient's cancer. The model can take the form of cultured cancer cells in which a sample of a patient's tumor is removed and cancer cells are extracted and artificially grown in petri dishes. Alternatively, the same cancer cells could be injected into a mouse whereby a tumor is formed in the animal. Under either circumstance, drugs are tested to see which one is the most effective in killing cancer. Historically, scientists use what are called established cancer cell lines. While cancer cells may grow well in the body, they don't grow long enough when taken out of the body. In order for scientists to have access to cancer cells for research, the cells had to be modified for long-term growth. To achieve such long-term growth, the cancer cells either spontaneously change or had to be genetically modified to grow forever. Those resultant cells are called established cell lines, which can then be used indefinitely for testing cancer drugs. However, established cancer cell lines suffer one major disadvantage. They are so far removed from the patient's original cancer that they no longer are representative of the patient's original cancer biology. So why do cancer drugs pose significant shortcomings regardless of the type of drug developed? The reason may be analogous to the parable of the blind man and the elephant. In the parable, a group of blind men touch an elephant to learn what it's like. Each one feels a different but single part of the elephant. The blind men then compare notes and discover that they are in complete disagreement in describing an elephant. This parable illustrates the different perspectives of human judgment, implying that one's subjective experience may seem true, but his or her experience is inherently limited by its failure to account for the entire reality. From a historical perspective, cancer is viewed differently from the prism of the chemist, the vascular biologist, the virologist, the immunologist, and the geneticist. Each scientist attacks the problem from the vantage point of his or her background and training. Cancer is mistakenly viewed as a homogeneous group of cancer cells, meaning they're all the same. In reality, it is not, as illustrated in the current figure. Cancer consists of a variety of different types of cells. There are multiple types of cancer cells with different behaviors, not just one type. Cancer cells possess multiple genetic mutations. There are cancer stem cells, which I will discuss later, their blood vessel cells, immunological cells, tissue fibroblasts that produce connective tissue. The type and amount of connective tissue combined with the blood supply to cancer define the material properties of cancer. That is, whether the tumor is hard, soft, rubbery, and whether it has a rich or poor blood supply. And this property is important in terms of how well a drug can penetrate a tumor. Thus, understanding drug efficacy and resistance is incomplete without considering the entire population of cells that make up cancer. Cancer stem cells are one of the major determinants of drug resistance. We generally regard stem cells as being a good thing, but they also have a dark side. If a stem cell becomes cancerous, it becomes an important determinant to the fate of drug resistance. You can think of cancer like a family tree where the oldest living ancestor, represented by the cancer stem cell, repeatedly differentiates or gives rise to a new generation of cancer cells. Each generation then gives rise to another generation of cancer cells. At each generation of cancer cell division, the lifespan of that generation grows shorter. Thus, the last generation of cancer cells may be the most susceptible to the chemotherapeutic effects because they lack an inherent capacity to divide and replace itself with new daughter cells. The extinction of these late generation of cancer cells from chemotherapy can now release the brakes to allow early generation of cancer cells to take off and grow because there is less competition for the nutrients in their microenvironment. In this scenario, chemotherapy manages to kill off all the progeny but not the cancer stem cell. At some later time, some trigger results in activating the cancer stem cell, which results in completely replacing all generation of daughter cancer cells. The take-home message is that the cancer stem cell is more resistant to chemotherapy, and failure to eradicate it may result in cancer relapse, drug resistance, and even cancer metastasis. Just like bacteria that show different susceptibility to antibiotics, cancer also exhibits different susceptibility to chemotherapy. As depicted in this figure, cancer cells are grown in two-dimensional structures in petri dishes, like tiles on a floor. The cancer cells are then exposed to the surface of these cells to a variety of different standard and non-standard chemotherapy, from low to high dosages. As shown, drugs that are represented on the far left are the most potent, while those represented on the far right show the least activity. Now, there are two attractive features of this test. First, and most obvious, one can identify which drugs a patient's cancer would best respond to. Second, and perhaps less obvious, is that one can directly measure the dose of chemotherapy that kills a patient cancer and compare that drug concentration to the calculated drug concentration from the prescribed dose. If the effective measured concentration that kills cancer exceeds the calculated concentration, then it is highly likely that the patient's cancer is going to be resistant to that chemotherapeutic agent. On the flip side, if the measured drug concentration that effectively kills a patient cancer is far below the calculated drug concentration, then the patient's cancer could likely be susceptible to that agent. Thus, this direct approach of testing cancer drugs on a patient's cancer outside of their body may provide predictability on whether a chemotherapy, individually or in combination, will be effective without exposing patients to unnecessary costs and risk of ineffective drugs. However, tumors are not two-dimensional structures but have three-dimensional architecture. If we take the most potent cancer drug from the prior example and deliver it to the same cancer cells but now are arranged in a three-dimensional tumor, we see a very different result. We see that only those cells located on the outer perimeter are susceptible to chemotherapy, leaving the internal cancer cells unharmed. Also, the larger the tumor, the more difficult it is for chemotherapy to penetrate and kill cancer cells. In this example, a small molecule was used and showed limited ability to kill cancer because it can't penetrate or the drug is not sufficiently potent at the center of the tumor. If this is observed with a small molecule, one can only imagine the challenges that could be observed with even larger molecules like proteins, genes, and cells that need to penetrate tumors to reach the area of interest. So in summary, while targeted therapies have been touted with much hype and promise, they are not on the immediate horizon in displacing conventional chemotherapy. Each patient's cancer has a unique biology. Cancer is more complicated than just genetics or immunology. There are a variety of cells that make up cancer, and cancer biology is governed by the interaction of all of these cells, not just a few. Drug resistance is dependent in part on cancer stem cells, the intrinsic drug activity, and how well drugs can penetrate a tumor to exert its effect. From this background, the Institute is focusing on the following research activities. We view cancer holistically as a complex tissue engineering problem. To solve this mystery of cancer, we are disassembling all the parts of the cancer, the stem cells, the cancer cells, the vessel cells, the fibroblasts, and the immunological cells, and will attempt to reassemble the parts to better understand how the entire tumor governs drug efficacy. We are also developing strategies to predict whether the standard administered dose of chemotherapy will kill a patient's cancer. We are interested in finding more effective strategies to penetrate drugs into tumors, and we are also interested in finding more effective strategies in limiting drug toxicity to patients with the notion that fewer side effects will result in better patient outcomes, greater survival, and lower healthcare costs. In our final session, we will discuss the Institute's Translational Clinical Research Program in Oncology, Predicting Clinical Outcomes to Chemotherapy. I thank you for listening to this program. Remember, your donations go towards supporting this medical research. JP2MRI is a 501c3 nonprofit that specializes in finding cures and better therapies exclusively using a variety of adult stem cells and cancer cells. JP2MRI is one of only a handful of pro-life medical research organizations that does not support or participate in any embryonic stem cell research. The Institute devotes over 55% of each dollar spent on scientific research, while most national not-for-profit medical foundations devote much less of their expenditures to actual research. Further, the Institute is entirely focused on reducing the time and cost to get therapies through the regulatory process. To learn more about JP2MRI, go to our website at www.jp2mri.org and consider a donation. Also, tell your family, friends, schools, and church groups and social media networks about the Institute.